Good morning. Just confirm me, am I audible? Just confirm me, am I audible? Basha? Okay, fine. Let's start today's class. Today we are going to start a developmental biology. Okay, particularly we have to focus on the process, spermatogenesis and oogenesis process. Okay, without any delay, we'll start our class. See, this is somewhat related to the process of spermatogenesis. Here, what will be the stage? Spermatogonium. What is spermatogonium? See, when it comes to the process of spermatogenesis and oogenesis, you should know these kind of terminologies. What is sperm? What is spermatozoa? And what is spermatid? And what is secondary? And what is primary spermatocyte? Like that, you have to aware of the terminologies. Okay. See, what is spermatogonium? So you should know whether the spermatogonium is in the diploid or in the haploid stage. So the spermatogonium will be in the diploid stage, and this will be undifferentiated diploid stem cell. Okay, and that will be present in the seminiferous tubules. Okay, spermatogonium is a undifferentiated diploid stem cell that is present in the seminiferous tubules. And where will be the exact location? Basal lamina of the seminiferous tubules. Okay, and primary spermatocyte will be this spermatogonium. As it is diploid in nature, it is undergoing a cell division, particularly the mitotic cell division. Okay, as a result of the mitotic cell division, the spermatogonium has been converted into primary spermatocyte, which is also in diploid in nature. Okay, see that too in the near basal lamina of the seminiferous tubules. Okay, in case in the secondary spermatocyte, this results in the primary spermatocyte. See, this will be the meiotic division of the primary spermatocyte. Now, as a result of the meiotic one division of the primary spermatocyte, it results in the two haploid cells. So obviously, see this 2n will give rise to 2n, that is primary spermatocyte. Spermatogonium under the process of mitosis, it is gives rise to again a diploid primary spermatocyte. And once again, this primary spermatocyte is undergoing a meiotic one division. As a result of meiotic division, as it is a reduction division, this diploid is getting converted into the haploid, two divided into two haploid secondary spermatocytes. Okay, and where exactly all this division is getting taking place in within the seminiferous tubules. Okay, now early spermatid. See, this is the result of what is this early spermatid will be the result of meiotic 2 division of the secondary spermatocyte. Right, see this meiotic 2 division of the secondary spermatocyte that results in the 4 haploid cells. Okay, that is also in the lumen of the seminiferous tubules. And late spermatid will be the differentiated spermatid with their head, midpiece, tail preparing to become a sperm. So from this late spermatid, we are going straight to a well-developed structure of the spermatozoa or the sperm. Okay, see the spermatozoa will be the fully mature sperm cells. This cells has the capacity to fertilize the ovum, okay, which is present in the female reproductive tract. And see this sperm will be released into the lumen of the seminiferous tubules and eventually pass to the epididymis. This will be the reproductive structure present in the male reproductive part. Okay, this is what exactly the process of spermatogenesis is. See here, the exact location will be the seminiferous tubules of the testis. And here, what are all the division is actually taking place here means it was mitosis, meiosis 1 and meiosis 2. Okay, and what will be the result of this process of spermatogenesis? Here we are going to produce spermatozoa, that is which is in the haploid in nature. This haploid haploid sperm, we are getting this haploid sperm from the diploid spermatogonia. From this diploid spermatogonia, it has been through the process of mitosis, we are going to get a primary spermatocyte, which is also in diploid in nature. And once again, it is undergoing a meiotic II division and gives rise to two secondary spermatocyte. And once again, it is undergoing a meiotic II division. Once again, it gives rise to four haploid secondary spermatocytes, four haploid spermatid. Okay, and as a result, each one is gives rise to an individual sperm. Okay, that's all about the process of spermatogenesis. See, as I said before, spermatogonia, which is in diploid in nature, under the process of mitosis, once again it gives rise to a diploid primary spermatocyte, and under the process of meiosis 1, it gives rise to two haploid secondary spermatocytes. So from this, you have to make a note of it. The primary spermatocyte is diploid in nature, and secondary spermatocyte will be in the haploid in nature. Now, from the secondary spermatocyte, under the process of meiotic II division, it gives rise to four haploid spermatids. Okay, this is what a spermatid, and after that, see, from spermatids is converted into sperms. What is that? See, the spermatids is converted into sperms, and that process is said to be as the spermiogenesis. Here, the sperm is getting synthesized. Sperm genesis means synthesis. From the spermatids, we are going to synthesize a sperm. So that process is said to be as the spermiogenesis process. Right? Now, oogenesis. Here initially it was in the it was like an oogonium, that is diploid in nature, that is considered as a diploid stem cell. Okay, this undergoes a process of mitosis and produce primary oocyte. Where exactly this particular primary oocyte is getting produced? In the ovarian cortex. In the primary oocyte, see this oogonium undergoing a mitosis arrested in the prophase 1 until puberty. Okay, so this is the result of oogonium, which is undergoing the mitosis. Initially, oogonium, it is a diploid stem cell. Okay, now this stem cell, definitely there has to be undergoing a division, and that division will be the mitotic division. As a result of this mitotic division, this oogonium going to produce a primary oocyte. Right, now this result, the oogonium, it undergoing a process of mitosis, and now here it is arrested in puberty, arrested in the prophase 1 until the puberty. Okay, now that is happened in the primordial follicles, that is within the ovarian follicles. Now the secondary oocyte, this is the result of primary oocyte. We are going to produce the secondary oocyte from the primary oocyte by the process, by the cell division meiotic 1. Okay, by producing a polar body and a haploid secondary oocyte. See, this primary oocyte is diploid in nature, that is 2n. It is undergoing a division called meiotic 1 division. As a result of a meiotic division, it is going to give rise to two haploid cells, and in that two haploid cells, one will be the polar body and one will be the haploid secondary oocyte, and where that is happening in the Graafian follicle. Now, as a result of this haploid secondary oocyte, once again it is undergoing a division called meiotic II division. As a result of fertilization, it is going to produce another polar body. Okay, and this has been released from the Graafian follicle to the fallopian tube. So this particular thing will be happening in the fallopian tube. Okay, so where it is going to happen? In the ovaries. Okay, what are all the stages involved here? What are all the cell division taking place in the process of oogenesis? Means mitosis and meiosis 1 and 2. Okay, and what will be the outcome? You are going to produce an egg, also the polar bodies from one primary oocyte. See, oogonia differentiate into primary oocyte during the fetal development. These points are very important. Kindly make a note of it. Here, see, this oogonia differentiate into the primary oocyte. It was differentiated into the primary oocyte that was happening in the fetal development. Okay. Now, this primary oocytes arrested in the prophase 1. It does not undergo any of the stages after the prophase until the puberty. Okay, each menstrual cycle typically releases results in the release of one secondary oocyte during the ovulation. So during the menstrual cycle, that is monthly, once this secondary oocyte is getting released, okay, that process is said to be as the ovulation process. At that point of time, one secondary oocyte is getting released. Now the ovum is released from the ovary if fertilized. See, it is fertilized, the ovum is released from the ovary and completes the cell division, that is meiosis 2, to become a mature egg and capable of fertilization. This is what the process of oogenesis is. Okay. Now, see, we have oogonia, that is stem cell, which is diploid in nature, undergoing the process of mitosis. It is going to give rise to further, it is undergoing a division. As a result of the division, further it is going to give rise to a diploid cells, and those diploid cells will undergoing a meiotic 1 division. As a result of that division, it is converted into primary oocyte. This primary oocyte, it is not completing its division, but it is stopped up to the prophase one, that will be up to the puberty. Now this primary oocyte is undergoing a division called meiosis 1 division. As a result of this division, it has been gives rise to one polar body and one secondary oocyte, and the secondary oocyte is the one further undergoing a meiotic 2 division. Once again, it gives rise to the second polar body and the one of the other haploid cell, and that is called as the ovum. Okay, this is what a process of oogenesis is. Now, answer for this question: where does spermatogenesis primarily occur? That will be seminiferous tubules. What is the chromosome number of the spermatogonium cell? Spermatogonium will be diploid in nature. Which process reduces the chromosome number during spermatogenesis? Whether it will be the meiotic 1 or 2 or mitosis or spermiogenesis. In the meiotic 1 division. See, in the meiotic 1 division, you should know that in mitosis, when it comes to mitosis, always it gives rise to a diploid cell. As a result of a division, each cell gives rise to a diploid cell. Okay, but meiotic division is also called as a reduction number. Why we are calling that as a reduction number means because there will be a reduction in the number of chromosomes. So obviously in the meiosis 1, we are going to get we have a reduced number of chromosomal number. Okay. What is the product of meiosis 2 in spermatogenesis? Give me a correct answer. That will be a spermatid. Meiosis 2 produces a spermatid. And from the spermatid, we are going to give rise to a sperm. And what that process called as? Spermatid gives rise to spermatozoa or sperm. What will be the name of that process? What will be the name of the process? That is what? Spermiogenesis. Okay. Where do spermatid mature into spermatozoa? Where will be the location? Please be on time. See, I have explained the concept. Just go through the recorded. Okay. Yeah, that will be epididymis. Okay. What will be the main function of spermatozoa? What is the main function of spermatozoa? See, if you answer quickly, we have some baggage in yesterday's class. Evolution, we have to go through the question papers question. Okay. So just answer me quickly. The main function of spermatozoa, what will be the function? It will be carrying a genetic material to the egg because it is the one going to fertilize the egg, right? Which hormone stimulates spermatogenesis? That will be the follicle stimulating hormone. Very good. And what is the approximate duration of spermatogenesis in humans? Spermatogenesis process duration. Just think whether it will take weeks or it take months. Is it 3 months? Spermatogenesis takes place approximately 64 to 72 days to complete in humans. Very good. Where are Sertoli cell located and where is their function in spermatogenesis? What is the function of Sertoli cells? See, when it comes to the process of spermatogenesis and the process of oogenesis, hormones plays an immense role. Okay. That will be in the seminiferous tubules. See, the function of the Sertoli cells, it has to nourish the sperm cells. So, what are all the nutrients required by the sperm cells that has been delivered by the Sertoli cells? Okay. Which of the following is not a part of a mature spermatozoa? In the mature spermatozoa, acrosome is not a structure. See, the question is wrong. Okay. Don't worry about the question. Here they are saying that acrosome is a structure on the head of spermatozoa that contains enzyme for penetrating the egg, but they are considering this is not a part of a mature spermatozoa, but actually this is a part. Okay. Where does oogenesis primarily occurs in females? Very good. That will be in the ovarian cortex. Now, which cell in the ovary gives rise to oocytes? That will be oogonium. Oogonium is the one differentiate into oocytes during fetal development. At what stage do primary oocytes arrested until puberty? That will be prophase 1. Okay. Now, what triggers the release of secondary oocyte during the ovulation? That will be the luteinizing hormone. This is the one triggers the ovulation. Ovulation means what? Release of the secondary oocyte from the Graafian follicle. So this is the hormone which influences this particular process, right? Now, what happened to the first polar body after oocyte maturation? What is going to do? What is the purpose of having the polar body and what is their function? It disintegrates and it is reabsorbed. Okay, it is not functional and all. Which structure surrounds the oocyte during ovulation? The most important question and famous question. During ovulation, which structure surrounds the oocyte? Very good. That will be zona pellucida, which is made up of a glycoprotein. It is the one that surrounds the oocyte. Which hormone maintains a corpus luteum and progesterone production if pregnancy occurs? Important question. Human chorionic gonadotrophin. This is the hormone which maintains the level of corpus luteum and progesterone production early in the pregnancy. Okay. How many mature ova are typically produced during one menstrual cycle? That will be only one. Okay. For each menstrual cycle, only one ovum is going to produce. Okay. Just keep in mind that. What is the fate of secondary oocyte if fertilization does not occur? That will be, it is expelled during the menstruation. Okay, this is what a fate of the secondary oocyte if fertilization does not occur. See, the secondary oocyte along with the sperm, zona pellucida and some follicular cells is expelled during the menstruation if fertilization does not occur. Correct? Now, which structure provides nutrients and hormonal support to develop the oocyte? That will be granulosa cells. This is the cells which provides the nourishment and hormone. They are going to support the developing oocyte within the follicle. Which process produce genetically diverse gametes? Genetically diverse means what? That will be the intermixing of two uh, different traits between two different chromosomes, and that will be obviously it is meiosis. Why we are having a genetically diverse gametes in the process of meiosis? Just tell me what will be the phenomena which is taking place in the process of meiosis. Proper division is not occur. See, I have explained you linkage and crossing over. Crossing over, because of the crossing over happened in the process of meiosis, we are getting a diverse gametes. And what is happening in the crossing over? This crossing over is taking place in which stage in the process? In the meiosis. Answer me quickly. Okay, these are all the basic question. At which stage this crossing over is taking place? You should say in the packaging stage. Okay. What is the function of Leydig cells in the male reproductive system? No, no, that's so okay, fine. Just knew it. Okay, no need to make excuses. What is the function of Leydig cells in the male reproductive system? Leydig cells is going to produce a testosterone. So this will be an important question. See, Leydig cells, on the influence of this particular luteinizing hormone, they are going to produce a testosterone, right? Now, which hormone regulates the menstrual cycle and promotes the development of secondary sexual characteristics? That will be estrogen. See, this is the hormone regulates the menstrual cycle and promotes the development of sexual characteristic features. See, if I asked which is regulating the pregnancy, then you can say the progesterone. Okay, but this will be the menstrual cycle that will be completely under the control of estrogen. What is the approximate duration of menstrual cycle in humans? I think I have given you the flowchart regarding the menstrual cycle. I hope everybody has read that. Duration of menstrual cycle in humans. That will be 28 days. Which structure releases the mature ovum during the ovulation? See, this is the structure. Fallopian tube is the one. This is the one releases the mature egg, that is secondary oocyte, from the during the process of ovulation. Now, what is the fate of mature sperm after ejaculation? Ejaculation is nothing but a release of sperm into the female reproductive tract. And once it reaches the female body, what is the fate of mature sperm?  'Sperm'. 'Sperm' Why option D? See, it is somewhat unrelated to the question, right? See, it has to fertilize the egg. That is the duty of the sperm, correct? Which hormone is responsible for triggering ovulation in females? Was luteinizing hormone, right? What is the purpose of acrosome in spermatozoa? There will be a lot of, see, this acrosome will be the modification of which organelle? How can we say that? Lysosome. Sorry. Which cell organelle, which this acrosome will be the modification of which cell organelle? That will be the Golgi complex. Very good. Obviously, it have a lot of enzymes. And what will be the one particular enzyme which is going to break down the structure of ovum during ovulation? During the sperm, when it reaches the ovum, what is the name of the enzyme? Very good. Hyaluronidase enzyme. Okay, good. And here the purpose of having the acrosome will be fertilizing the egg, because it has to break the outer layer of the sperm and it has to release the enzymes. The enzyme which is present inside the acrosome, which helps the sperm to penetrate into the egg by breaking the egg because of the enzyme called the hyaluronidase. Which structure is responsible for the production of seminal fluid? That will be a prostate gland. Very good. That contributes the production of seminal fluid, which nourishes and transports the sperm. Now, in which phase the menstrual cycle does in which side which phase of the menstrual cycle does the process of ovulation has been occur? In the ovulatory phase, no doubt in that. Okay, that's all about that. Wait, let me show you. Okay, just answer this question. In yesterday's class, we have stopped in this slide. Which of the following is not concerned with the speciation process? So this will be your PYQ, and if you know what are all the things we have discussed in the class, you can answer this question. Here, two identical population genetically diverge to a point at which their subsequent merger will not occur. Pre-mating isolating mechanisms promote divergence of population. Okay, see here, pre-mating isolation that results in the divergence. That was before mating. Okay, before mating there will be a certain isolating mechanism that results in the divergence. This is what the statement is saying. And option C will be post mating isolating mechanisms ensures the population divergence. Okay, you have to know whether the pre mating isolating mechanism or the post mating isolating mechanism whether which of these two isolating mechanism it promotes the divergence, which is that possible, and mechanism that promote gene exchange between the populations. And you have to find whether which of the following is not concerned with the speciation process. And that will be option B, mechanism that promote gene exchange between the population. Okay, what are all the other questions we have? Evolutionary relationships among the group of organisms are termed as, and this question we have discussed many times. We have discussed many times in our class during non-car data and car data. The word I have been used to that very often. And what is the term? Yeah, that will be phylogeny. Okay. So in evolution, we have caught up with less number of questions. There will be no much questions from evolution, maybe from ecology. Okay, just answer this question. Hardy-Weinberg law. Hardy-Weinberg law assumes. So this is how your question will be. You should know the conditions, whether it will be the larger small population size or small population size. And when it comes to the random mating, this Hardy-Weinberg law prefers random mating or a selective mating, non-random mating. Whether there has to be the migration or no migration, whether there has to be the mutation or no mutation. Okay. And that will be larger population size, prevalence of the random mating, absence of migration, and absence of mutation. See, you people have been saying option B. Do we study anything about survival of heterozygotes? Do we study about that word, survival of heterozygotes? We have taken into a consideration regarding the population, random mating, mutation, and the migration, and one more thing will be gene flow. Okay, there will be no point of having a confusion. Okay, that we doesn't read any of the word like survival of the heterozygote. So this will be your answer. Okay, just be careful when answering the question. Even if you know, you have to be very conscious while answering the question. I think that's all will be the question from the evolution. Okay, yeah, that's all for tomorrow's class. We are your topic will be fertilization, cleavage, gastrulation, formation of germ layers, and parthenogenesis. The second line of your syllabus from the development